So if we could get started on this afternoon's sessions. So I'm glad you all managed to find your way back and hopefully got something to eat. Today we're starting with the speaker who I happen to know personally the best. As I mentioned this morning, first thing, I spent about 4 years in his lab. And he is also a fellow of the AAAS, so we have 3 of the 4 speakers. And was recently the president of the Society for Neuroscience. And unlike many societies, this one has 38,000 members. So it is quite a job to take on. He's well known for his seminal work in mapping the structure and function of the brain, which, in fact, Terry showed some of his early work from in 91 with Brodmann. And has been doing a lot of work trying to figure out useful, valid ways to amalgamate the enormous amounts of scientific data, neuroscientific data that there are. As you may know, every brain is different, so it's a difficult problem trying to figure out exactly how to take all of the data that we have and align it to infer useful things. And so he has been developing ways of blowing brains up. In fact, most of these are here. Blowing brains up in the bottom left, flattening brains in the top right, and trying to have sort of machines do things like detect cortex, like you see on the top left. No doubt he's going to treat us to the kinds of insights that we can gain from doing these sorts of manipulations. And his talk today, excellent, the title has not changed, is The Structure, Function, and Development of Cerebral Cortex. Join me in welcoming David Van Essen.
Thank you, Chris, and I must say it's a great pleasure to be here, albeit a bit belatedly. I'm sorry to have missed the morning session owing to my passport oversight habit that I just developed. But I'm here, and I also want to say, just as by pre-introduction, how much I enjoyed interacting with Chris in the time that he was at WashU and our paths overlapped. I found it remarkable that an individual with the kind of engineering and philosophy background that he had could dive into computational and neuroscientific issues with the energy and perspective that he did and come up with the contributions that started there and continue here. But my focus will, as you heard by the introduction, be on the cerebral cortex, and really I'm hoping to cover a lot of ground. Can you all hear me okay in the back? Okay. I want to cover a lot of ground, and I will show variants of this. Well, easy come, easy go. There it goes. Of this outline to show you where we're heading. It'll cover quite a bit of ground, but there is some continuity across themes, cross-cutting ideas, and I hope by the end you'll feel you've gotten something for your money relating to many, if not most, of these topics. So, maybe that's the handedness that works. So I'll talk about human cerebral cortex. Individual variability, as Chris pointed out, is hugely important and a dominant theme of the talk. It'll lead into questions about how the cortex develops and how it folds, and how we can use insights from that to build and make use of better human cortical atlases and map data of various sorts onto the cortical surface and move forward in mining data using new tools that I'll describe. I'll also show you how I, as a basic neuroscientist, very hardcore for several decades, have now moved into a collaborative arena looking at issues of scientific interest, but also of significant clinical relevance, and I'll talk about specific questions of looking at ways in which the cortex is abnormally folded in disease conditions. Then I gather I missed some discussion relating to comparisons across species, and I'll certainly be talking some about how we can use our what we call surface-based atlasing tools to make comparisons across species, particularly between monkeys and humans. And I'll come back to at the end with some comments about how these tools are now getting more out there in the field and can be used by everyone to mine data off the web, and I'll give you some examples of that. So the work in my lab involves actually, these days, not so much experimentalists in the lab, but rather a team of talented computer programmers, Donna Durker, John Harwell, and Ping Gu particularly, and a talented technician, Aaron Reed. And you'll be seeing the fruits of their labors as well as the fruits of some collaborative work coming forth. It may be helpful to start by orienting you to that wonderful thing, the human cerebral cortex, as seen by structural magnetic resonance imaging. And if you take a slice through the brain and make a movie going from back to front, these are a series of coronal slices. And I'm sure most, if not all of you, have seen at least an image or two like this, where you can see the left and right cerebral hemispheres, gray and white matter, and the cerebellum underneath, and then the complexity of this folding pattern as we go from back to front and then from front to back. It's just a wonderfully complex, convoluted structure. We have, over the years, developed tools to segment the cerebral cortex, that is, to capture the shape of the cortical folds, and the outcome of that in this particular example is the same movie, but if we stop, now you can see that the pinkish regions are the core of the segmentation volume for the left and right hemispheres, and then the cerebellum underneath. So this is one particular individual, and having carried out that segmentation, we can then ask the computer to wrap a surface around the segmentation. So this is one slice of that segmentation of a different brain, of just the right hemisphere, and then we capture the shape by generating a geometric tessellation surrounding those folds, including the deep convolutions, and then we paint that surface with, in various ways. The one that's illustrated here is what we call a Sulcal depth map. So we can inflate this surface to a highly smooth shape, but preserve information about what it originally looked like by shading the surface darker where the sulci were deeper. And basically each point on this surface, we compute the distance from down there to this outer, what we call the cerebral hull, to create this Sulcal depth map that's a continuously varying indirect measure of shape. We can, one more sort of introductory movie to orienting yourself to the cerebral cortex. This is an individual hemisphere. We're looking at the lateral view of what we call the fiducial surface. There's a hole there where there isn't cortex, that's non-cortical medial wall. We inflate the surface but preserve the Sulcal depth map. And we can make cuts, as you would make cuts in the Earth's surface to make flat maps. And we can flatten this out like a pancake. Or, perhaps a better analogy, if you look at the two hemispheres, each one of them is the equivalent of about a 13-inch pizza inside your skull. The analogy is further useful because these pepperoni-like analogs here are in the ballpark of the size of individual cortical areas. And we'll come back more seriously to this question of what is the cortical mosaic look like. Well, yeah, those are wormholes. The uniqueness. Everyone in this room is special, not just to your mother and your grandparents and so on, but you are unique individuals physically in many ways, but your personality is really dictated by the wiring of your brain. And to paraphrase Bill Clinton's, it's the economy, stupid, for thinking about brain function. It's all in the wiring, the connections. I'm going to have less to say about individual differences in connectivity because we still don't have a whole lot of data on that front, but I'll be emphasizing issues about the individual variability just in the overall size and shape of the brain as it fits in the skull, a lot about differences in folding patterns, and some about differences in the size of these individual areas and how each area relates to the nearby folds that sometimes form landmarks. So if you take structural MRI images like the first one you show, look at two individuals. Now these are sliced parallel to the midline, the parasagittal plane. There's the eye, there's the cerebellum. And you can see that indeed skulls are shaped differently, as you can see externally looking around the room, and the brains fit compactly inside the skull. They have a different overall shape. They're also different in their orientation as they lie in the scanner. Some brains are long and skinny, some are more quasi-circular, so substantial differences in shape. And then substantial differences in the specifics of how the cortex is folded. Folding of the cortex gets a large surface area squeezed into the compact skull and gives it a large surface area. But if you look in detail, this is our first but not the only pass through this issue. Here are two individual hemispheres viewed from the medial side. And you can recognize, at least if you have some experience, you can recognize several standard sulci. The calcarine sulcus here in one individual is also present in another individual, but whether you look at it on this original 3D surface or on a slightly inflated surface, you can see it's really folded rather differently. And ditto for other sulci. The cingulate sulcus is a long groove in all cases, but the details of these local wrinkles are quite different. On the lateral aspect, two different individuals. Here's a nice long superior temporal sulcus groove that's been slightly inflated. Here it breaks into two different trenches, it furrows. So these are not small differences, and your brains, your cortex is different from one individual to the next as much, if not more, than your fingerprint patterns are different. I think of these as cortical fingerprints in a way. And so that diversity is obvious then at just macroscopically looking one way or another at the pattern of convolutions. The issue of how the convolutions relate to the cortical areas that we'll be talking about is best understood for the primary visual cortex shown here. So back in the occipital lobe, if you take a coronal slice of a postmortem brain and stain it for Nissl substance or for myelin, you can see, particularly in this myelin stain at a glance, that there is a stria of Gennari that is very powerfully demarcates the full extent of V1. When Andrews et al. In the Purvis lab looked at this systematically, they were able to precisely calculate the surface area of V1 in many individuals and compare that to the surface area of the calcarine sulcus. And they noted, as been known for more than a century, that V1 is mostly in the calcarine sulcus, but it extends outside along the margins. And when they plotted calcarine sulcus area against V1 area, what they found was that there's a more than twofold variation in V1 surface area and almost as much in the calcarine sulcus. So the point is there are correlations, but it's far from perfect, even in this notably, relatively well-behaved domain. One more drive this point home example taken from a different study, Amunds et al. In the Zillis lab, carefully reconstructed the extent of area V1 in another group of individuals analyzed post-mortem. And you can just see that the area V1 is not just in the calcarine sulcus that extends out to the occipital pole, but to a considerably varying degree for each individual. So when you think of an area like V1, which has a very precise topographic map of the visual world onto the surface here, and you think of where is the very center of the gaze, the fovea represented, it's going to be somewhere back here, but it will be at physically different locations in the brains of different individuals. So the idea of correspondence of one cortical location, one little cluster of neurons in one individual to another, is a complex concept. You have to think of it in terms ultimately of function, but typically we only have the folding pattern to help us out with this. So these are issues that arise just in looking closely at individual human brains and cerebral cortices. The issue gets compounded further when you step back and say, well, what about other species? And it's rather extraordinary how not just that other brains are complex in their folding, but the diversity in the detailed pattern of folding, and even in the degree. The poor old manatees, extremely lissencephalic, dolphins and others are as if not more convoluted than us, but in patterns that vary a whole lot. So how do you make sense of all that? Well, that's going to get us into a set of issues of cortical development and folding, the next phase here. And the questions are, when and how does folding occur? How are folds related to functional areas? Why the dramatic species differences? And later on, we'll come on to the question of whether folding abnormalities may reflect connectivity differences. But a starting point comes from a collaboration that's been underway for a couple of years that I've gotten involved with a couple of pediatric neurologists at WashU, Terry Inder and Jeff Neal, and students in their orbit have been in the interesting circumstance of access to a whole pipeline of preterm infants that are born in St. Louis. They're kept in the intensive care unit at Children's Hospital there, and with proper approval from the relevant entities, they're able to do MRI scans on these infants at various ages. So here is a coronal slice through a 25-week-old preterm infant. And you can see at a glance it's not very convoluted at all. And as these scans go from 25 to term equivalent almost at 37 weeks and then compared to a 4-year-old, it's getting more complex. But in order to apply the kinds of analysis that I've already shown you in adults, we needed to be able to segment these infant brains at all of these stages. And to cut a long story short, the segmentation algorithms that we have that work fine on adults don't work at all on the infants. And so Jason and a co-student or fellow student, Andy Knudsen, developed their own algorithm that works nicely, and you can see we can get segmentations and surface reconstructions of these same infants, and you get the power of a surface-based visualization of the complexity as it evolves of these folding patterns. And in some of these infants, we can actually start longitudinal studies, in effect, able to scan the same infant at 29 weeks and 33 weeks. And you can see how much the cortex is expanding in complexity just within a month. And we can make a movie to make it more visually graphic to see the progression between these two ages. And we can also see at a glance that the expansion in cortical surface area over this 1-month period is not simply uniform expansion. There's actually appears to be more expansion in some regions than others, and that's something I'm actually quite interested in, and we hope to learn more about how consistent that is, given this one example. There are also clear dramatic cases of pathology in some circumstances. Here is an infant with post-hemorrhagic ventricular dilatation, essentially a form of hydrocephalus early on, and you can see that infant has a much different, more lissencephalic cortex than the age-matched equivalents. And we can quantify these, not just look at it by visual inspection. So this has emerged into a major funded collaboration because the clinical relevance is actually rather strong. There's a very high incidence of clinical problems from cerebral palsy through epilepsy and autism and other disorders that occur in an unfortunately high fraction, up to 40 or 50% of the very preterm infants. And it's not known why. Why are they at so much more risk when they're out in the intensive care unit? So we hope to learn more that's of clinical relevance as well as just the fascinating developmental questions. But we know already that these preterm infants are not as mature as term equivalent infants in terms of their brain cortical folding at the matched ages. We know there are lots of pathologies in the cortex and in subcortical white matter, and one of the objectives is over a long term to identify correlations between behavioral abnormalities and underlying brain pathologies using the tools that you've seen. But there's also a very basic question that I touched on and now we'll delve into. Why does the cortex fold? How does it fold? And that's an idea, actually, I got into this a decade ago purely serendipitously, thinking about the question of what might cause cortical folding. The baseline facts are that the specification of cortical areas, the mosaic of cortical areas we'll talk more about, but we know that that occurs quite early in development while the brain is still very smooth and the cortex is smooth. We know also that there's a lot of variability in the size of different areas. I already gave you some of the evidence for that arise just because there's more proliferation of the population that gives rise to one area than versus another. We know also that the convolutions, about the time the convolutions are forming, is about the time when long-distance cortical-cortical connections, exemplified by the wiring diagram that Terry already showed you earlier. And so the idea, the light bulb in the middle of the night idea that came up was, maybe these wires are not just passive. Maybe they're actually generating mechanical tension and driving the folding process. And that's schematized here in this idea of tension-based morphogenesis in general and tension-based cortical folding in particular. That, for instance, think of area V1 and V2, particularly in the monkey. It starts out very smooth, but as the massive pathway between V1 and V2 in both directions forms, suppose that mechanical tension actually just draws those regions together, forming a gyrus in between them, and conversely a sulcus would form in regions that are maybe somewhat connected but not as strong. They lose out in the overall competition. And that general idea is still a hypothesis. It is unproven. There's a lot of evidence supported. I'm not aware of anything compelling against it. But it's a hypothesis, but it's one that shapes my way of thinking about a lot of issues that we've touched on and will continue to talk about. One of the points is that this one simple idea, tension-based morphogenesis, can dually account for both the consistency of cortical folding and the variability. So the idea is that consistent folding, you consistently see a calcarine sulcus and various others in regions that are dominated by relatively large areas and with robust pathways that are driving things pretty much the same way in each individual. Whereas variability may occur in what you can think of as balkanized regions where you have a mosaic of lots of small areas and they're bickering and fighting and the outcome is different from one individual to another. And so that's the working hypothesis. And then one corollary that I'll come back to is that abnormal connectivity in terms of the strength or pattern of connections could hypothetically be a cause of folding abnormalities in disease conditions. Okay, so moving along, let's now come back to the adult cortex, but use some of the insights that we talked about to think about better ways to make brain atlases. So an atlas is a representation of structure that provides a spatial framework and usually, though not all, or commonly, a coordinate system, and on that atlas one can put lots of reference information about where the folds are, what the areas are called, and so on, and a repository of many other kinds of knowledge, and use that to visualize, analyze, and compare in a variety of ways. The challenge, as Chris brought forward in the introduction is, how do you register data from individuals to an atlas? Ideally, you want to get areas that are functionally corresponding into precise alignment. A fundamental problem is we generally don't know where those areas are, so we have to use the next best thing, which is some aspect of folding patterns or structure or geography to constrain the registration. Historically, there have been two main approaches, but with many variations on a theme in each. One we call volume-based registration, and one we call surface-based registration. And I don't want to get bogged down in the methodological details, but I do want to give you enough of a sense to know where the field started and where we're going. So classically, volume-based registration started with a famous atlas, the Atlas of Taylorach and Ternaux, often called the Taylorach Atlas. It was actually one post-mortem elderly French woman gave her brain to science, and it ended up as a series of pictures of drawings of the shape of the cortical folds in different slice angles. And for many years, the idea was to take an individual structural MRI and to register it to the Taylorach Atlas by changing the dimensions, essentially fitting it like a shoebox to match the approximate shape of the Taylorach Atlas. And that was far better than nothing, and it worked, got the field going forward for many years. There have been successive improvements of this volume-based registration approach, but still the reality is that even with modern techniques, if you take two individual brains that have been successfully registered to a target MRI atlas, like this one on the right, the folding patterns are by no means identical, and when you average a bunch of these together, it gets blurred because of all that residual variability. And that's that's at the heart of volume-based registration. So that's what has motivated us and other groups to explore and now start touting the merits of surface-based registration. So, but the starting point for us was to get our bearings on how we should go about this. Given the variability I've already talked about, and now we're looking at it again, here are lateral views of 6 individual right hemispheres. Here are the slightly inflated versions of these. And if we look more closely at the inflated ones, you can always see a central sulcus, pretty consistent in shape and general trajectory. You can always see a Sylvian fissure as well. You can always see a superior temporal sulcus, but it's often variable in its extent, and I gave you another example of that earlier. And then there are some regions like this occipitotemporal junction that are just almost randomly crumpled from one individual to the next. So we took all that into account, and after a lot of inspection and a lot of thinking about this, ended up with a strategy that is encoded as the PALS approach, population average, landmark, and surface-based. So the surface-based motivation is to capitalize on the topology of the cortical surface in driving the registration process. And the landmark concept is to use a small number, literally only 6 landmarks, the central sulcus, Sylvian fissure, Calcarean sulcus, and a few others, and to drive an individual to the atlas using a well-defined spherical surface as the substrate, the shape on which we do the registration process. So here's the individual sphere for this particular case, and we register the landmarks of that individual to a population average target that's shown over on the right. Here are all the individual contributing subjects. Here's the average set of landmarks. And here's the relatively modest deformations that occur to align the individual to the atlas. So the basic point, though, is to give you a sense of how it works, but it does work. It provides an atlas that we contend is better than anything else currently out there, and conceptually has some satisfying characteristics. So one way of starting to make use of the atlas is to take the individuals from the dozen individuals who contributed to the PALS B12 version of this atlas take all their individual, what we call fiducial surfaces, and average them together to form something we call the average fiducial surface. It looks sort of brain-like, but not quite the same as any individual, but it turns out to be a very excellent target for mapping data onto the atlas in a way that's not biased by the choice of one individual or another. We can also make prettier versions of the atlas, mildly and strongly inflated versions, to use as representations on which you'll be seeing a lot of data displayed, starting right now. So we took these individual 12 subjects and manually and laboriously and carefully identified all of the major sulci using a classic Ono et al. Atlas as a reference, and painted each sulcus a different color, and then registered all of the individual sulci to make a probabilistic atlas of the different sulci. And it succeeds in better registering these sulci, substantially, more or less twofold better than classical volume-based registration. That's one piece of evidence that this method is actually working, but it's not perfect. There's fuzziness in the colors indicating variability in the layout of the different sulci, whether you look at it in on the surface or back in the average volume. We can quantify aspects of variability in several ways. So one thing we can do on these average inflated surfaces is to take the individual sulcal depth maps that I had shown you earlier and just average all those together. And in regions where there's consistency, these are relatively dark streaks, and in regions where there's variability, it's grayed out or browned out in here. But we can quantify the amount of variability and show that by our registration algorithm, there are several regions of quite low variability, the blue in particular, and other regions that are hotspots of high variability, particularly in parietal and frontal cortex. We can also look at hemispheric asymmetry. There's been an enormous amount of work on hemispheric asymmetry, both functionally and anatomically. We've revisited that using these more sensitive tools that I'm showing you here. And if you look at the average sulcal depth map on the left hemisphere and the right hemisphere, you can see most notably that in the superior temporal sulcus region, it's darker here on the right than it is on the left. And the outlined green region is the domain where it's significantly different between the two hemispheres. And the basic point is we've confirmed and better characterized known regions of hemispheric asymmetry and identified other regions where there are asymmetries that had been less well characterized or appreciated. We can move on. So what I've shown you up to now on this PALS B12 atlas are characteristic and information derived from the individual subjects that contributed to the atlas. But now we want to start pouring the paint on from data that originates in different sources, starting with a century-old source of the classic Brodmann cytoarchitectonic areas. This is what the legacy he provided us was just a couple of drawings. This is still widely used as a reference frame, even though it is not the most accurate or modern architectonic map, but we felt it was worthwhile to transfer the Brodmann areas onto our atlas and provide a prettier painted version, which in the Caret software that we use allows you to highlight and quickly identify which area is which color. We can paint functional information onto the map. So here is a map of functional modalities showing visual cortex as occupying about the posterior quarter of human cerebral cortex, auditory, somatosensory, motor regions, large expanses that are provisionally just assigned to cognitive in general or to limbic in these purplish regions. We'll come back to functional specialization more, but this is just a first pass at getting oriented to the major domains. If we come back to the finer grain resolution that modern functional MRI provides, some of the best relevant evidence and data comes from studies of visual cortex. Roger Tootell and a number of other groups have mapped visuotopic or retinotopically organized visual areas back in occipital and occipitotemporal cortex, and from data that they generated or put onto their own map, we transferred the map of visual cortex from one of their surfaces onto our PALS Atlas surface and painted it slightly different colors, but you can see this map of a set of visuotopic areas. There are more recent studies, including one from the Swisher et al. At the Boston University group, mapping visuotopic areas and extending well farther into the parietal lobe than had previously been done. And we've mapped their individual data sets onto our atlas and are continuing this process of painting the atlas with good data when we can get our hands on it. Though getting our hands on it is still a challenging issue, it turns out. So that's one part of the story, mapping cortical areas. There are lots of other kinds of data we would like to put on. One set includes just functional neuroimaging activations of the type that you see illustrated here. If you have an individual subject looking at not apples and oranges, but apples and other objects compared to scrambled versions of those objects, you see in each subject some kind of activation pattern in the difference between these two signals. It's similar but not identical from subject to subject. It's typical, the common practice is to average the data across subjects and determine what's statistically significant. And when that is done, we can map these data from a functional volume, population average volume, onto our PALS Atlas surface and compare that activation to various other kinds of information, such as the visuotopic areas you saw before. And we can do that across multiple studies, even when those studies happen to have been registered to different standard spaces. It's a technical nuance, but an important one, that SPM software and WashU software and the so-called AFNI software, they're registering to different volume atlases. And what we did was just to take our PALS atlas and register those to each of the standard spaces and get more reliability so that when we compare motion selective activations from a study by Jim Lewis et al. And see that that activation, the hotspot is centered in area MT, we can compare it to the object-centered selective activations that I showed you in the preceding slide and see that that is, it was registered to a different atlas, but when it's brought into this common surface space, the activation is in a partially overlapping but mostly outside of MT. And then we can bring other kinds of studies that may seem like they're coming from out of the blue, but they're interesting to compare. So here, for example, Harold Burton and colleagues looked at somatosensory evoked vibrotactile stimuli in blind patients, activates, lo and behold, visual cortex in a big-time way, as well as somatosensory areas. And here is the activation in these early blind subjects, including MT and nearby regions. So there's all sorts of cross-study comparisons that we're in progressively stronger position to analyze. Resting state functional connectivity, MRI, is a big mouthful, but it's turning out to be an absolutely fascinating, and it looks like it's going to be an incredibly powerful way of probing the functional organization of normal as well as diseased, disordered brains in a sensitive way. And it starts by telling a subject, go into the scanner, stay awake, maybe fixate on a spot, don't do any task. It's not hard work, just sit there. And then, while they're sitting there quietly, if you look at one region or another, any one small region of so-called voxel, and record the fMRI signal, the bold activation signal, if you look on a very slow time scale, this is 100 seconds here, you find that at every single voxel, the signal's going up and down and up and down, not oscillating, just varying. And if you look at nearby voxels, or sometimes distance voxels, the correlation, like between yellow and orange, is incredibly strong. And in other places, it's actually anti-correlated. And what this is discovered by Biswal a decade ago, but Mark Rakel and colleagues at WashU and a number of others are really digging this as quite a gold mine. And I'll give you a few highlights here. One pattern that emerges is the so-called default network, a region that is strongest, has the strongest signal when a subject is not doing a task, and then it's weaker when you activate or have them do cognitive tasks. But in this case, the pattern of the default network is revealed just when the subjects are sitting there doing nothing, and you find these greenish and bluish regions essentially define the default network, and they are anti-correlated with the red and yellow regions that tend to be areas that are activated in other circumstances when a cognitive task is done. Another component of this is it's not just all about cerebral cortex. That has been my major fascination, but the cerebellar cortex is a hefty chunk of convoluted tissue as well, and it is much harder to work with from a surface-based approach because the cerebellum is much thinner, and you don't get enough. You saw it in that first movie. You can hardly see gray and white matter in the cerebellar cortex, so you cannot routinely segment cerebellar cortex. Won't happen soon, but maybe someday. But in the meantime, we did laboriously and accurately segment and get a surface of one individual, the colon cerebellum. And so we now have a hybrid PALS population-based cerebral atlas and a colon cerebellar atlas that coexist. You can look at them concurrently, and you can paint data from the same, for example, this default network that I referred to. You can see where it lights up with strong correlations of cerebral cortex, but also a portion of cerebellar cortex. And so we're going to try to get the cerebellum more onto the map using this approach. Here's one other example of the dorsal and ventral attention systems that Mike Fox et al. Studied. And the dorsal attention system has a correlation pattern that's rather similar to the cognitive activated network that I mentioned before. The ventral attention system is predominantly on the right hemisphere of lateral parietal and inferior frontal cortex. But it was defined by task-related attention studies, but here it is showing up as a subnetwork within using this resting state connectivity approach. An even more striking one that's a bit complex, and I'm going to just whiz through it because the punchline is this may be incredibly powerful in teasing apart individual cortical areas in individual subjects. And this is from work by Alex Cohen and others in Steve Peterson's lab, and we've collaborated with them to start by taking a grid of points on a surface, a flat map here. And for each point here, the computer cluster is asked to grind through and identify a map of these resting state correlations with every other voxel in the entire volume and generate a map, and then to do the same thing for another neighboring seed point and compare how similar those maps are and to generate a similarity map, basically. So these white regions, the functional correlations are not shockingly, very similar for each of these points in this domain. But then the correlations fall off as you go farther away. The striking thing is that the correlations don't fall off gradually. They fall off sharply. And if you compute using an edge detection algorithm, you can identify sharp edges in this particular pattern. And if you do the same analysis for every point in this domain, you end up with the punchline here is a pattern that shows streaks of sharp transitions bounding darker regions, which are where the correlations are relatively consistent. The hypothesis is that these will, at least some of them, will identify individual cortical areas. Pat, you had a...
What area is that?
This is in the medial anterior cingulate cortex. It has not, the critical test is, does this correlate with areas as, for example, have been revealed by the Johansenberg and Oxford group using DTI? Work in progress, answer anxiously awaited. But if it works, it's going to be very exciting. Data mining. You all are by now, I presume, used to PubMed as a way of saying, for instance, if you want to find out who's doing what or what's known about fMRI studies of memory, well, you can find a lot of studies that give you an answer, and in many of these cases, you'll go to a publication, link to it, and find pretty pictures, and then boring, dry tables of stereotaxic coordinates that give the spatial coordinates for activations or deactivations. And the literature is rife with this. In fact, these two tables here, the punchline is there are about an estimated 6,000 studies, neuroimaging studies, that report the stereotaxic coordinates. And the slope is still going up, so it's probably 1,000 or more per year of data coming in. And how do you make sense of it? It's very tough to just run from one table to another or look at one picture and another that's in a different format. So what we've taken to is a project that has now got enough traction. You're seeing some of the early results here. Aaron Reed in my lab has patiently and laboriously gone through every single issue of Journal of Neuroscience, identified the 360 studies that report a total of 13,000 stereotaxic coordinates in these tables, almost all of them. And here they all are pulled out, extracted, and put into the caret-compatible format so we can display them on our PALS cerebral and Collins cerebellar surface. And then there's a whole lot of metadata that are linked to these representations. One point just right off the top, the Journal of Neuroscience is not biased for any one question or topic, and so it's kind of interesting that the density of activations, if you take the density of these points and just map them onto the surface, there are some real hot spots of activation in the anterior insula, region of MT, and the medial cingulate, anterior cingulate. And we can relate those very specifically, not just to anterior insula, but to area IAL of the insular cortex by Joel Price's partitioning scheme. And he has some real ideas of what that area is doing. I'm not going to go into that there, but rather to say that this is one level of first-order data mining that is, to some extent, it reflects investigator bias. A lot of people want to study MT, but it may also reflect something about what turns different parts of the brain on. In any event, we can dig a lot deeper using search tools such as this from our caret software. You can just load all these foci, these data, and then search for regions that are activated based on their location, what area they're in, journal keywords, or other functional tasks, and start getting beginnings of a more comprehensive survey of what things turn on one or another region of the brain. And so all this is going towards a 21st century way of data mining this giant neuro-Dagwood sandwich of many kinds of information stacked conceptually on top of one another and available in an atlas framework. I'm going to start whizzing through because time is flying, but I want to say some about these developmental-related disorders. We serendipitously got into a collaboration with Ursula Bellugi and others who had a population of Williams syndrome subjects. And Williams syndrome, if you're not aware of it, is not an inherited but a genetically based disorder. Hemideletion of chromosome 7 leads to an unusual constellation of severe deficits such as in spatial memory and other things that are actual strengths, musical interests, hypersocial behavior. And there have been studies to look at the brains and have found abnormalities in the brains of Williams syndrome, but we felt there was perhaps more to be mined. If you look at the structural MRIs of 2 Williams versus 2 controls, it's hard to see any difference at a glance. If you look at surface reconstructions of 2 Williams and 2 controls, it's still very hard to pick up anything. But we had a toolkit that we, in fact, partly developed in order to answer these particular questions. And the seeds for it you've already gotten an idea of already. If we look at the average sulcal depth in Williams syndrome compared to controls of the left hemispheres here, they don't look identical. And if we actually subtract one from another, you can see there are hot spots of substantial differences, but they also tend to occur in regions of high variability. So we had to get a handle on what's signal and what's noise, and we did that using statistical tests to generate a t-statistic for the depth difference at each point, and then use the size of the clusters to decide what is convincingly real. And the upshot of it is, in this paper published 2 years ago, there are 33 identified what we call folding abnormalities in the Williams syndrome cortex that span all of the lobes, but a swath going from dorsal posterior to ventral anterior, where in some cases it's deeper in the controls, some cases deeper in Williams syndrome. And one of the reasons I'm fascinated by this goes back to the question of, can we relate a macroscopic structural abnormality, a folding problem, to something of more neurobiological direct interest? And maybe so. The best example to plant the seed is the olfactory sulcus, seen in this ventral view of the orbital frontal cortex, on average is rather deep in controls, and on average it's rather shallow in Williams syndrome. And you can see that in individual subjects, so I won't belabor that here. But it plants the seed of saying, maybe in this region, the primary developmental abnormality is that the cortical areas, particularly 13B and 13M that Joel Price has identified as lining the banks of the olfactory sulcus, maybe those areas are physically smaller in Williams syndrome, or maybe they have scrambled connections. In either way, the tension-based cortical folding idea would say normally you get these gyral grooves and a sulcus in between based on the connectivity patterns of these areas, and maybe the folding, the mechanical tension is working out differently because the wires are scrambled or fewer in number. It's a testable hypothesis, but that awaits future studies. One other disease-related study to mention briefly, in a collaboration with Christine Wu Nordahl and David Amaral's group, we looked at cortical folding abnormalities in autism, a much more prevalent and concerning disorder, as you're probably all aware. Autism is not a single disorder. It's known to be a spectrum, but what the underlying neurobiology is has been a puzzle. When we look at 3 different categories, low functioning, high functioning, and Asperger's, basically we found folding abnormalities in each of these subgroups, but they were strikingly different. Left side only in the frontal or perculum region in low functioning, more towards the parietal cortex in high functioning, and way back in the interparietal sulcus in Asperger's. And just, I'll leave it at that, but this approach of sulcal-based or surface-based morphometry is proving to be a powerful way of looking at some intriguing clinical issues. 2 quick riffs through the remaining topics. Comparisons between monkeys and humans are of obvious interest, monkeys being such an intensely studied animal model. We know a huge amount about architectonic areas in monkeys. Here's a map of a moderately well filled out mosaic that Jim Lewis did. probabilistically generated from 5 subjects and turned into an average that we can view and work with in Caret and identify whichever area we're interested in. But there are many competing partitioning schemes. There's no consensus for most regions of monkey cortex, and this just illustrates that many of the competing schemes from Ungerleider and Desimone and others that we and others have done have been brought into our monkey atlas framework, so at least you can see where the differences are and where the commonalities are, and that in itself is a step forward towards deciphering what's going on. We know not just about the existence of cortical areas in monkeys, but we know a lot about the connectivity, not just a wiring diagram, but in some cases we have real quantitative data about the pattern of connections in individual monkeys from tracer injections in one or another area. And of course there's a huge amount known from single unit studies that is schematized in this hierarchical scheme that shows different levels of processing and different processing streams, and I'm just going to leave it that there's a lot known about that. And in addition, there's a growing amount known from functional MRI that can be carried out in monkeys, including very recently studies using this resting state connectivity approach that I mentioned. It works not only in humans, it works in monkeys, it works in anesthetized monkeys. So it's not a corollary of some conscious state, which is an interesting point on its own. And this pattern of functional connectivity, the correlation pattern seen in this resting state analysis, turns out to have strong correlates with known connectivity patterns, part of the wiring diagram. And I'll leave that as a sidebar, and I'll just jump into the monkey-human comparisons then by saying if we've got evidence for these correlation patterns and even hints that there may be a default network in monkeys analogous to that in humans, it would really be nice to be able to make careful interspecies comparisons despite the dramatic differences not just in size but in the overall layout of the cortex and in the, for instance, the amount of visual cortex in the two species and its relation to cortical folds. How does one go about that? Well, basically, we contend that surface-based registration approaches are vital for this, but in this case we have to ignore the sulci and use whatever functional landmarks we can get our hands on, such as V1 and V2 are identified in both species, and auditory cortex and MT, frontal eye fields, and several other areas that are identified. Just notice at a glance that MT and A1 are very close to one another in the monkey. They're very far apart in humans, a hint of the differential cortical expansion that we're going to see evidence for in a moment. We can take functional MRI studies using the same task, objects versus scrambled objects. You saw that in humans. It's been done by Brodmann's group also in monkeys. And we can use the commonalities in fMRI studies to constrain the registration. And that turns out to be important. Whizzing through the details, basically, we identified a set of 23 landmarks based on functional criteria that we are moderately, reasonably confident of in monkeys and in humans. We project those onto spherical maps, and we deform the monkey sphere to match the human sphere, and then we get pretty pictures as a result, including a movie that shows the expansion between monkey and humans to give you a sense of what we think is going on. And then we can generate areal expansion maps, which is our best estimate of what regions have expanded the most, these hotspots of expansions, and what have expanded the least. And you can see occipital cortex is rather unexpanded, and its anterior lateral prefrontal cortex and this parietal, lateral parietal regions that are our estimates of the hotspots. And we can compare those with various other kinds of data. I'm going to just whiz by this and just say this is a work in progress to relate these evolutionary patterns or trends that we're getting evidence on to the many other kinds of information from fMRI studies in humans, from connectivity studies and physiology studies in monkeys. So we think that we're entering an interesting age for more objective and careful comparisons of species differences, but I would emphasize strongly that identifying convincing homologies remains a tough, challenging problem. Lastly, I'm going to, in about 2 minutes, whiz through about 20 slides that will give you a flavor of what you can do in terms of accessing most of the data that I've shown you here. Most of it is actually available on the web, tied into the SumsDB database that we've generated and the WebCarrot software visualization package that lets you look at the data without downloading any data or software unless you're ready to and want to. And it's not just a database in its own right. We put a lot of effort into linking online journals and databases in ways that are mutually beneficial and informative. And so one example is if you start on the SumsDB homepage and just click on our Human Atlas, you'll quickly launch WebCarrot and you'll see a default view of the PALS Atlas. You'll have a set of pre-configured scenes that you can choose from, and you can choose by description whichever you're interested in. If you want to see a particular collection of areas, you can just jump right to a map of visuotopic and orbitofrontal areas, and it's interactive. You can just click on any point there on the atlas surface and say, what is that? Oh, it'll highlight that, and it'll give you a lot of information, including this is area 47R, and if you want to say, well, who said that, you can go to further information that links you to the journal article that made that claim, and you can go up and read the Unger Ferry Price article and see their map on their individual atlas surface that was the source of this map on our PALS atlas. So that's going from the database to the atlas. We can also to the online article. We can also start with online articles. A growing number of online publications have data in the database, and you can read about it when you read the article and say, oh, the data are available here. Let me click on this link. It takes me not just to the database and go figure it, but right to the source of that data, those data. You click on, if you want to actually download it, you can, but you may want to just launch WebCarrot by this route and immediately bring up a repeat of the scene that was in the publication. But this is now not an image, it's an interactive atlas that's being displayed in this WebCarrot software. It repeats individual scenes, and it lets you go beyond the scenes and drill down into the data set. And you may want to see more than was in the scene, different views, different underlying data. It's all, in many of these cases, it's there. And in one last example, you can take the Williams syndrome data set that is also in the atlas. You can see different views of it, including some that aren't in the publication, and you can compare some of the location of some of these folding abnormalities to maps of architectonic areas that have been generated by the Carl Zillis's lab, and see what you think of the relationship between a structural folding abnormality and somebody's architectonic partitioning scheme that is mapped onto the atlas but also readily accessible on their original online data. So I hope this gives you a feel for the kind of exploratory environment that modern neuroinformatics is starting to provide, that lets a user sit at a computer terminal and, I wouldn't say effortlessly, but rather efficiently nowadays, migrate between databases, software on your machine, online journal articles, and learn more faster and have more fun at it. So thanks for your attention.